Welcome to the President's Dream Colloquium, Understanding Medical Marijuana. My name is Zamir Punja. I'm a professor in biological sciences. I'm the chair of the organizing committee. If anybody of you have any questions, comments, concerns later on, please come and see me. It's my distinct pleasure right now to introduce our president and vice chancellor of Simon Fraser University, Andrew Petter. He's going to come give you some welcoming remarks and give you a little bit of context on the President's Dream Colloquium. Thank you very much, Andrew.
Well, thank you very much. Thank you very much, Samir. It's great to see this turnout for the inaugural lecture in the spring 2017 President's Dream Colloquium. You know, when we set up the President's Dream Colloquium as an idea, as a dream, it very much came out of our vision to be an engaged university and to provide another forum for engagement on campus to bring students and faculty and staff together to really interrogate issues that matter. And I must say it's proved to be very, very successful because the dreams have been the dreams of faculty organizers who have thought of great speakers, great topics, and have brought together students to study, to learn, and of course enable the rest of us to come and partake in the lecture series. So it's proved to be something that I really value, and I love the opportunity, and I hope you do too, to benefit from the hard work that's been done to bring fabulous lectures to us on topics that really matter. I understand this semester we have 18 students who are enrolled in the Dream Colloquium. These are students from all sorts of disciplines: health, psychology, biology, communications. They enroll for credit in the colloquium. They prepare papers. They learn from the speakers. But what's great is because of the interdisciplinary nature of the students, they learn from each other as well. Former students have told me that they really appreciate this. They see it as a sort of like a field school on campus. It gives them a chance to relate to speakers who come from near and far and to engage with students they otherwise wouldn't get a chance to engage with. The whole idea is to foster interdisciplinary dialogue, to promote advanced research, and to create a conversation. And I'm just delighted that we have this semester a chance to look at issues around the theme of medical marijuana. And as I look down the list of speakers and what's happening in Canada, I suspect you might even get into some issues around recreational marijuana, with Anne McClellan and some others coming. While marijuana is known for its medicinal and recreational properties, there is, I know, much research still to be done to better understand what those properties are, what its applications are, certainly in the medical context. And with Canada moving to legalize marijuana, this is a perfect time to become informed about marijuana, about its properties, about its medical applications, as well as issues that may affect us in respect of legalization generally. And so I view this colloquium as a real opportunity to learn about an important topic in a way that is much deeper and richer than we would otherwise be able to do. Now, the reason we have that opportunity is because some faculty members some time ago decided to devote a lot of time and energy to putting together the program that we're going to enjoy today and over the succeeding weeks. And I really want to acknowledge and thank them. They include our colloquium chair, Zamir Punja, Peter Rubin from the Department of Biomedical Physiology and Kinesiology. Zamir, by the way, is from the Department of Biological Sciences. Dr. Neil Watson from the Department of Psychology. Dr. Urs Ribéry from the Department of Psychology. Neil Boyd, the School of Criminology, and Cora DeBeck from the School of Public Policy. So themselves a pretty impressive interdisciplinary group. And the students who are enrolled, you're very lucky to have those professors in addition to the speakers that we're bringing in as people from whom you're going to learn over this semester. I also want to acknowledge the work of the Office of Graduate Studies that does the administrative support for the colloquium, including Gladys Weed and Dina Coburn, Dean Wade Parkhouse. Their help in making this colloquium series and other colloquia possible has been just fabulous. And of course, this afternoon, I'm going to get out of the way because we all want to hear from our guest speaker, Dr. Ethan Russo, and I want to thank him for his time and effort to come here. And like you, I'm looking forward to his discussion on the pharmacology of cannabis. So, a perfect way to start off, another great colloquium a wonderful opportunity to say thank you to those who've organized it, and to look forward to the opportunity as well to share your ideas and questions in this lecture and the ones that follow. And with that, I'm going to turn things back over to Zamir to present an overview of the colloquium series and to introduce our guest speaker. Zamir?
Thanks, Andrew. When I applied to the President's office to fund this colloquium, I really wasn't sure where it was going to go. But as you can see, SFU is an engaged university. They want to reach out to the community, and they went ahead and gave us the green light. And so here we are today to talk about this particular topic, which is extremely relevant. As Andrew pointed out, there are a number of people that were involved in organizing the seminar series. They're all listed here, extremely diverse members of the organizing committee from public policy, criminology, biomedical physiology, kinesiology, and so on. Special thanks go to Gladys. Gladys I categorize as 3 E's: efficient, energetic, and enthusiastic. So Gladys, thank you very much for your help with putting all of this together. As well as Carmen. Carmen organized many of the posters and banners that you see here, and Jennifer was involved in helping us organize and select gifts for the speaker. So thank you to all of you for your valuable contributions. So as you know, we're here to talk about this plant, cannabis, and we'll spend the next 10 weeks or so, for those of you that are involved in the course, studying various aspects of this plant. And I'm a plant biologist by training, and I've worked with various plants over the last 30 years. And I can tell you right now, this is probably the most fascinating and most interesting plant when you compare it to things like tomato and potato and other plant species. I could never get an audience like this for any type of colloquium that deals with those plants. So it is interesting, and it reaches across boundaries. It reaches across various boundaries that relate to health, that relate to medicine, that relate to criminology, public policy, business, entrepreneurship, product development. The list goes on and on and on. It's the only plant species that I know of that can relate to so many different disciplines. Hence, we see a great turnout of people to understand this in more depth. We have speakers and sponsors, and these are some examples of one of the gifts that we are going to present to them that acknowledges their involvement. Let me just give you a quick overview of the speakers that are going to be part of this colloquium series. There are brochures going around, and certainly you can find this on our website. Today, Dr. Russo is going to talk about the pharmacology of cannabis. It's a great place to start because that's where all the chemical molecules are found, where all the molecules are interacting with each other to give you these medicinal and potentially recreational properties. Dr. Page will speak in 2 weeks. He's at UBC, and he also runs Anandia Labs, and he'll talk about the genetics and the genomic applications of cannabis. We'll have Dr. Philby come 2 weeks after that, who will talk about neuroscience and potential addictive properties of marijuana, which is also an area that's extremely interesting. Mark Ware, many of you have heard his name, will also speak to the clinical applications and health applications of cannabis. The Honorable Anne McClellan, who was the chair of the Task Force Committee recommendation report, we're honored to have her join us in March, and she'll talk about the task force recommendations, how they derived the process, and so on and so forth. And then finally, Dr. Kilmer will speak to us about cannabis legalization and the experience that they've had in the US and how it could potentially apply to Canada. So you can see a very, very diverse range of topics going from science to medicine all the way to public policy and other issues. Again, just to remind you, for those of you that haven't had a chance to see this document, this is the task force report. It's worth reading. It has some very important recommendations for Parliament and for the Prime Minister to consider. It's a fairly easy report to review, but it does have some important recommendations. And it was released about 6 weeks ago and certainly has created a lot of interest. The other report I'd like you to be aware of is one that was released not too long ago from the US National Academy of Science Engineering and Medicine that is also entitled The Health Effects of Cannabis and Cannabinoids, the Current Status of Evidence and Recommendations for Research. In both the task force and this report, one of the very strong recommendations is for continuing research to allow investigations into claims of health properties and various other aspects. I'll just read you the first sentence from the summary of this report that says, "This is a pivotal time in the world of cannabis policy and research. Shifting public sentiment, conflicting and impeded scientific research, and legislative battles have fueled the debate about what, if any, harms or benefits can be attributed to the use of cannabis or its derivatives." So clearly this is an important time, not just for Canadians, but also for people that live in the US. I have some dreams as well, regarding the colloquium at least, and I'd like to share those with you so that we have an understanding of what the goals and objectives were for this colloquium. The first being, of course, to provide you as an audience, those of you that are here in this room, those of you who are watching on the webcast, an evidence-based assessment of the opportunities and limitations to the use of cannabis. The second dream that we'd like to accomplish is to provide you with a forum for discussion. Discussion with us, discussion with the speakers, discussion amongst yourselves on this particular topic. To provide opportunities to enhance collaborations. Collaborations not just with SFU, but with UBC, with Kwantlen, with University of Victoria, with various other academic institutions so that we can further the understanding of cannabis, provide a learning opportunity for students, and have them more informed as a result of the debate. And finally, of course, to engage and take advantage of opportunities, not just on campus, but off campus as well. So I'd encourage you to think, to talk, to debate, to discuss, and to engage. And that would be a dream for us if all of that was to happen. And of course, SFU, as Andrew pointed out, has given us this opportunity, a very unique opportunity, I might add, to increase the public engagement on cannabis. And so before I end, I'd like to acknowledge sponsors, of course, the President's Office, the Office of Graduate Studies and Postdoctoral Fellows, and our Faculty of Science. And in addition, we receive funding from NSERC through a Connect grant, which allows academics to interact with industry and with various stakeholders. But in addition, there are a number of companies and organizations that are listed here. And if you're wondering how we ended up with so many names on this list, I basically went up to them and said, would you be interested in sponsoring this colloquium? And they all said, yes, we can. And yes, they did. And so here we are with a list of organizations that are clearly involved in furthering the education process, in helping you and them understand what the applications of cannabis might be. So we thank the sponsors, all of them that are listed here. You will be meeting representatives of these sponsors, and I hope you can continue discussions and engage with them on this topic. We have a website. I think many of you know what it is. And if you don't, it's www.sfu.ca/medical marijuana. The talks that are presented here will be live streamed and webcast. So if there are sessions that you can't make, we encourage you to go to those. Those of you that are listening on webcast, we encourage you to come physically to the campus and meet people and listen to some of the presentations. The last two lectures, the one by the Honorable Anne McClellan and by Dr. Kilmer, will be downtown, at the downtown campus. So the first four are going to be here in Burnaby, and the last two will be downtown. So you can get more details on that. We're also going to have a meet and greet networking reception after each of the lectures. So as you head out the doors at around 5:30, make a left for the food, make a right for the cash bar, and we'll hope you'll stick around and discuss with your colleagues. And with that, I'd like to introduce our speaker today, Dr. Russo. Many of you know him or have heard of him. He is very, very well recognized in the field of developing knowledge about the various ingredients that are found in cannabis. His training was from the University of Pennsylvania in psychology, where he then moved to the University of Massachusetts and did his medical training there before doing residencies in pediatrics in Phoenix, Arizona, as well as in Washington State. He was a clinical neurologist in Montana for 20 years, and it was at that time where he was exposed to cannabis as potentially a product that could be used for chronic pain management. and he's been working with GW Pharmaceuticals for many years since. He's now in a consulting role and works with various other organizations. He's had faculty positions in pharmaceutical sciences at Washington, has been visiting at the Chinese Academy of Sciences, and has held various roles in cannabinoid societies and medicines. There is a list of publications on the website. There are a few that I've listed here just to indicate that there are current articles coming out in 2016 and 2017 that are quite relevant. Articles published in the Trends in Pharmacological Sciences, Frontiers in Pharmacology, the British Journal of Cardiology, and so on and so forth. So I would encourage you to try to get hold of some of Dr. Russo's papers and follow those for more information on this particular topic. And with that, thank you, Ethan, for coming up this way to visit us, to talk about, I know, what is one of your favorite topics, and inform our audience. Thank you very much.
Thank you. Well, it's a pleasure to be here to lead off the President's Dream Colloquium, also to be north of the 49th parallel. So I'm just going to take a moment to find my presentation. My email is listed there, ethanrusso@comcast.net. I would be happy to hear from you and help provide you with materials related to this talk or other topics. So it all begins with the plant. I will not talk about marijuana. That is a pejorative Mexican slang term for the plant of the old world, properly known as cannabis. Sativa means cultivated, and it indeed is. It's been a camp follower for humans throughout the centuries. The primary medicinal portion is the unfertilized female flower that we see in the upper left. If we look a little closer, the real medicine is produced in what are called capitate glandular trichomes. Those are the clear spheres on top. And these produce a variety of cannabinoids and terpenoids that we'll discuss in detail. Specifically, THC works on something called the CB1, cannabinoid 1 receptor, because its activity resembles endogenous molecules within us, anandamide and 2-arachidonoglycerol, and that's how it exerts many of its effects. So there is the plant. I agree with Zamir. It's the most fascinating plant I've ever run into. It's probably the most useful single plant on Earth, because if you were stuck on the proverbial desert island, it could provide your medicine, your food, your fiber, and even your fuel, as it was so used in times past. This is a close-up of the trichomes, and the clear sphere is where the cannabinoids and the terpenoids, the aromatic components, are stored. At the base, you'll see the cells where these are produced. But again, there's much greater production in the flowers compared to the trichomes that are on the leaves, which are about 1/100th the volume and actually produce different sets of chemicals that all perform an ecological role for the plant. Keep in mind, as much as we'd like to anthropomorphize, this plant is about 60 million years old evolutionarily, whereas we as a species are something more like 600,000. So it wasn't put on God's green earth to make us high. It had its own reasons for being, and we've just helped along the way. You'll hear a lot from Jonathan Page about this sativa versus indica business, and to me, I've been interviewed on this topic. I think it's mainly nonsense, the way these terms are bantered about in common parlance. There's debate about whether they're multiple species or a single species. Not to sound reductionist, but I'm interested in the chemical content of the individual plant. But we do have the problem of how you classify them. We're interested in the shape, how tall it is, what the leaves look like. more importantly, the specific cannabinoid and terpenoid content, but also for consumers. What does it smell like? What does it taste like? And most importantly, what are the effects when it's administered? This is one scheme for doing this. I particularly endorse this approach. You'll see on the right what's called a phytofax. This is now a patented method of classifying cannabis, and you see that it will give numbers on the cannabinoid content, the terpenoid content, the moisture. Then in the next panel, you've got a picture of the bud, the ratios of the two primary cannabinoids, and then you've got pictures of plants. In this instance, you've got a little bit of lemon and pepper. It sounds like a marinade, but that is because the main terpenoid components are familiar to us from those particular plants, limonene and caryophyllene in this instance. Also, you get a depiction of how it will smell, how it will taste, and then on the right, what the effects are as decided by consumers. This one is good on energy and things like that, and low on sedation. So we get down to the bottom panel and we see why. The yellow is limonene, which we'll discuss as a strong antidepressant. Beta-caryophyllene is a very powerful analgesic that synergizes with the cannabinoids. What we don't see a lot of is the predominant terpenoid in most chemovars of cannabis. Chemovars are chemical varieties. That's a proper term for what people call strains. But there's almost no myrcene, and this is directly the opposite of what we usually find in cannabis of commerce currently. But we're not going to confine ourselves to the glandular trichomes today. Rather, we're going to look at all these plant parts, and each of them has its own peculiar biochemistry, much of which can be harnessed for medicinal purposes. So while we're primarily interested in the unfertilized female flowers, where up to 30% of the content can be THC and other cannabinoids, you've got these other forms. And we'll talk about roots, which are probably unfamiliar to most of you and are thrown away commonly, but they've been medicinally useful for thousands of years as well. And we'll look a little bit at the seeds and even the sprouts. So we'll start out with the phytocannabinoids. That means plant-based cannabinoids as produced in cannabis. And a lot of this material is in a publication I hope to submit on Sunday. Jihan Marku and I were asked to do this for Advances in Pharmacology. Hopefully that'll be out later this year. But we're going to talk at varying length about each of these 10 cannabinoids. So there's not just one called THC. To look at the biochemical production in the plant, it starts with these two precursors, geranyl pyrophosphate and olivotolic acid. In the plant, the cannabinoids are initially produced as acid forms, cannabinoid acids. And depending on which enzyme is involved, you can get the precursor of THC or CBD. But it doesn't become THC until it's decarboxylated, removing this CO2 from that group. But the plant, just go back for a second, you'll notice that these have a 5-carbon side chain, so these are called pentyl cannabinoids, and they're the most familiar. But the plant also can make 3-carbon side chains. From a different precursor, which is up here, divernic acid. Professor Mechoulam, who's the grandfather of cannabinoid science, calls this an example of nature's law of stinginess. I prefer another name, which is enzymatic substrate promiscuity, meaning that cannabis has ESP. And just to show all of them together, I wanted to point out one other thing, and that is that geranyl pyrophosphate is not only a parent molecule to the cannabinoids, but it's also the parent molecule to the terpenoids. So these are related. They're half siblings, and unlike some blended families, they get along really well. as we'll see. Of course, most familiar is THC, delta-9 tetrahydrocannabinol. This molecule was searched for about 150 years until Professor Meshulam on Gaoni in Israel positively identified its structure in 1964. It is a weak partial agonist at the CB1 and CB2 receptor, which is exactly the profile of anandamide. So it's working quite like it, but this is exactly different than synthetic cannabinoids like K2 and spice that you hear so much bad things about. These can be hundreds of times more potent and have totally different effects to THC, which is actually very weak in comparison, as it should be, to act physiologically on this system. It is analgesic, a painkiller. It reduces itch. It's a bronchodilator. In the 19th century, there were cannabis cigarettes for asthma. It's also a neuroprotective antioxidant. It helps prevent brain damage, and there's actually a patent from the US government on this, despite the fact that it's a forbidden drug, reportedly with no medical use. So we're into a lot of discrepancies south of the border these days. Additionally, not working through the receptor, THC has 20 times the anti-inflammatory power of aspirin and twice that of hydrocortisone. It's a muscle relaxant. That's the basis of its approval as a drug for spasticity and multiple sclerosis, an approved indication for the drug Sativex or nabiximols, approved in Canada in 2005, first in the world, but in 28 other countries. It is an antiemetic. That was the basis of the approval of synthetic THC, Marinol, in 1985 for treatment of nausea associated with chemotherapy. And of course, it's the primary psychoactive component. Now, unlike a lot of painkillers, like the non-steroidals, this doesn't inhibit COX-1 and COX-2. Drugs that do produce ulcers, heart attacks, and strokes as possible sequelae, and this does not have that liability. Additionally, it's been shown it can reduce beta amyloid, one of the toxic products that's involved in the pathophysiology of Alzheimer's disease. That was one slide on THC. We're going to require several on CBD, which may be less familiar to you. This is cannabidiol. This was isolated in 1940, but again, only positively identified in 1963. And it's been a mystery for a long time. It barely binds to the cannabinoid receptors, but it was known for a long time that even when it was present in a low concentration, it would interfere with THC binding. And it was only a couple of years ago that it was discovered that it was a negative allosteric modulator. It lodges on a separate site on the receptor and interferes with THC binding. Now, it doesn't knock it out entirely. Rather, it will limit the side effects when they're administered together. CBD is an even better neuroprotective antioxidant. It's more effective than vitamin C or vitamin E in this regard, and again, has a US patent. It is analogous to capsaicin, the active ingredient in chili peppers, as a TRPV1 agonist. Unlike capsaicin, which burns the skin, CBD is not caustic, but it has the same ability to downregulate the receptor, desensitize it, which is applicable to a variety of neuropathic pain syndromes. One of the main activities, particularly in regular usage, is it inhibits the breakdown of anandamide and thus makes it more available. Because it's working indirectly, it does not contribute to intoxication the way THC does. And it is an alerting drug in low and moderate doses. It is very prominent as an anticonvulsant. The preparation called Epidiolex has been through 3 positive phase 3 clinical trials in epilepsy and should be FDA approved later this year, I would expect. Again, unlike THC, it's an anti-anxiety agent. It has a property that's really interesting, and it shares with the other cannabinoids, and that is it is toxic to cancer cells at high concentrations, but at the same time being preservative for normal cells. So this is quite in distinction to conventional chemotherapy that's generally toxic, and this is why people lose their hair and slough their guts. So we have the opportunity here to have a drug that simultaneously kills cancer, is good for normal cells, is good for the gut, doesn't have these other side effects, allays nausea on its own, reduces anxiety. One could hardly hope for better. And then there's some other activities and other receptors like GPR18 that's involved in the pathophysiology of endometriosis, and that is something we surely should study in the future. For autoimmune diseases like rheumatoid arthritis and inflammatory bowel diseases, it antagonizes tumor necrosis factor alpha. Most of the drugs that are on the market for that have bad side effects. The biologicals used this way, and CBD does not have that adverse event profile. Again, not a COX-1 or COX-2 inhibitor. It also has turned out to stimulate the serotonin 1A receptor. This was work that I did with my colleagues at the University of Montana in the last decade. And that turns out to be the basis of its use as an anti-anxiety agent, but also is the mechanism of action as an antiemetic, combating nausea and vomiting, also reducing damage due to strokes and brain damage and hepatic encephalopathy and liver failure. It also works on the adenosine A2A receptor that is probably germane to its effects as an anti-inflammatory and painkiller. Something that we haven't been able to test in the clinic, it works on prions, slow viruses. If there were another outbreak of mad cow disease, this would be worth a try. And an interesting one, because this is a Schedule I drug south of the border, but it stimulates bone healing after fractures. And it could be that athletes will need to get this after their injuries in the future. Now, there are a lot of misconceptions about CBD, so much so they felt compelled to write an article about it. A lot of dispensaries will get excited about having a small amount. Although CBD is a very versatile molecule, it's not very potent. You do need a good amount, generally at least as much as there is THC, and sometimes a lot more. It will commonly be said that it's not psychoactive. It is. It's anti-anxiety, it's antipsychotic. What it is not is intoxicating. So this is a semantic distinction that's quite important. Most people think that cannabidiol is sedating. What they're mistaking is the fact that many chemovars that have a lot of CBD are also very high in myrcene, which is very sedating, and hence has created this misconception. Another one we hear in the States is that it's legal in all 50 states if it's extracted from hemp refuse. This is actually not true. The Controlled Substances Act has an exception to the exception, so when you take a legal material like hemp stalks and chemically extract it, it's illegal again. Another one that was popular this year because of an article that used simulated gastric acid to show a conversion of CBD to THC, that doesn't happen in your body. It's been known in the lab for 70 years that you could do this, but even high doses of CBD administered to humans will not produce any THC levels. And you have no enzymatic equipment, unlike the plant, to make that conversion either. So this is a schematic from an article that's just out in Trends in Pharmaceutical Sciences, going from the plant to the trichomes, to the precursors, then to cannabidiolic acid. After it's decarboxylated by heat or light, you've got cannabidiol. That's what the crystals look like when it's pure. And your liver turns it into 7-hydroxycannabidiol. We don't know whether this is active or not. What you don't get is conversion to THC, except in the test tube. So. Now, we're going to move on to another one. Cannabigerol is what's in line before CBD and THC. Now, normally, the plant doesn't stop here. It's high throughput. It's going to go directly from CBGA, the precursor, onto the others. However, there have been mutations found. GW Pharmaceuticals has a plant that stops at CBG. 100% of the cannabinoids are cannabigerolic acid, which becomes CBG upon decarboxylation. So this is a GABA uptake inhibitor. It has muscle relaxant effects. It's actually stronger than THC or CBD. It's shown antidepressant effects in animal testing. It will mildly lower blood pressure and intraocular pressure. An interesting application is it reduces keratinocyte production in psoriasis. It could be used that way. A really fascinating one is it has strong antiseptic and antibiotic properties, including MRSA. That's multiply resistant Staph aureus. There was an epidemic of that in the States in hospitals some years ago. But the idea of using cannabis as an antiseptic might not fly in all areas. And there's some effects on the alpha-2 adrenal receptors and on the 5-HT receptors. How these really work in humans, we don't know yet. There really haven't been any clinical trials, but this is a molecule that needs a lot more exposure. It also works on several of the TRP channels, could be applicable to pain on that level. And it seems to be a very, very powerful anti-anxiety agent without sedation. Another one that you probably haven't encountered is cannabichromene. It's found in small amounts in most kinds of cannabis, but this one is formed by a recessive gene. Both THC and CBD are produced by codominant genes. Normally, this is only present in small amounts and sometimes in younger plants. It is an anti-inflammatory. It's also analgesic, but less potent than THC. It has, again, some antibiotic and antifungal properties. It usually scores very well in comparison to CBD as an anti-cancer agent. This has also shown antidepressant effects. Again, works on various TRP channels. And it has some ability to inhibit Pha like CBD does and could boost anandamide levels. So this deserves further investigation. One of those propyl cannabinoids with the 3-carbon side chain is tetrahydrocannabivarin. So if you had 2 more carbons, this is THC. This is present in small amounts in cannabis plants from southern Africa, things like Malawi gold, Durban poison. It is really interesting pharmacologically because it actually is a neutral antagonist at the CB1 receptor at low doses, and then at high doses it flips over to being an agonist like THC. This has been investigated as a drug for the metabolic syndrome and treatment of type 2 diabetes. It does produce some weight loss and has effects on leptin that's involved in hunger mechanisms, increases energy expenditure, and basically would be helpful for a number of metabolic changes in the increasingly obese populations of the Northern Hemisphere. It also is a pretty strong anticonvulsant. It's been good for neuropathic pain. Because it's a neutral antagonist, unlike rimonabant, the weight loss drug that was marketed briefly in Europe and then taken off the market because of all its side effects, inducing anxiety, depression, seizures, tumors, it doesn't have any of those side effects. Again, a predominantly THCV cultivar has been developed, where 92% of the cannabinoids are THCV. Its counterpart is this, cannabidivarin. So this is like CBD, but with a 3-carbon side chain. It inhibits diacetylglycerol lipase, which is a biosynthetic enzyme of 2-AG. Actually, that sounds like it'd be bad, but we don't know what this would really do on a long-term basis yet. This has been used in humans and is under investigation now as an anticonvulsant for seizures of partial onset, focal seizures. Right. Now, as I mentioned, the plant makes cannabinoid acids. So the parent molecule of THC is this, tetrahydrocannabinolic acid. This will be the form in fresh, unheated cannabis flowers. It is there because it's insecticidal. But in the laboratory, it's a strong anti-inflammatory, again, through the anti-TNF alpha mechanism. So this could be good for autoimmune diseases. An interesting one from way back is this is tested in rodents for its anticonvulsant activity, and it required a very high dose, 200 milligrams per kilogram. But clinically, we know from colleagues like Drs. Sulak and Goldstein that small amounts of THCA seem to provide control of seizures in children where they weren't getting it before. But we know that this molecule, it's confusing. Initial reports were that it didn't bind to CB1, but work done in Canada by Aaron Rock showed that it does bind with a pretty high affinity. However, it doesn't pass the blood-brain barrier, and we still need to figure out how it could act on seizures at all. This has proven to be a very effective antiemetic, good for nausea, and in very low doses. So its counterpart, the parent molecule of CBD, is cannabidiolic acid. So this will be the predominant phytocannabinoid in fresh hemp. It's a natural herbicide and will kill fish if in a retting pond where hemp is being prepared. This one does produce COX inhibition, but at very high doses, and probably isn't a big issue. It seems to be an extremely powerful antiemetic. It's working through that serotonin 1A receptor mechanism, and it's a couple of orders of magnitude stronger than CBD, and it may be that tiny doses are all that's needed. Additionally, based on the historical data, we have a lot of stuff from the Renaissance herbalists about using hemp to treat tumors. But nobody's really studied this formally so far, and that needs to be done. This one's interesting because it's actually pretty common in cannabis, and we know nothing about what it does. So this is called CBGM, cannabigerol monomethyl ether. If somebody out there needs a graduate student project, this is a good one to figure out what it does. And then there's cannabinol. Cannabinol is the non-enzymatic oxidative end product when THC breaks down. A lot of companies are getting excited about this because they have this old cannabis that otherwise would be thrown away. So they're talking about how great it is. Well, it's actually not that exciting. It's like THC, but it has about a quarter of the potency. It does have some antifungal effects, and it is mildly sedating, mildly anticonvulsant, and it does have antibiotic properties, but it really doesn't distinguish well compared to the others. Right. Now, we're going to shift into the terpenoids, and what you'll see on this slide is a bunch of familiar plants, lavender, lemons, and the reason is they all have terpenoids that are common to cannabis. The terpenoids have gotten short shrift in the past. It was expedient for certain governments to say that the pharmacology of cannabis was all about THC and nothing else mattered. However, on the other hand, we had consumers saying, well, I really like this kind, or, you know, at night I prefer this kind. But they'd say it was all about THC. And in most cannabis, if we test, it's going to be high in THC and high in myrcene and not very different. But again, consumers have felt that there have been these differences, and they are due to the terpenoid content. Pharmacologically, these are extremely potent. The numbers look small, but they are very meaningful. They have pain-killing properties, anti-inflammatory properties, mood-enhancing effects, and synergize with the cannabinoids. So this is a schematic of some of the ones we're going to talk about. These are the monoterpenoids, 10 carbon molecules. They look quite different to one another, but they have in common that they're hydrocarbons, sometimes with oxygen, no nitrogen, and they all have 10 carbons. The first I'd like to discuss is one that there isn't enough of these days. This is alpha-pinene. It's actually the most common terpenoid, monoterpenoid in nature. It's anti-inflammatory via the prostaglandin E1 mechanism. It's freely bioavailable. If you work in a sawmill, you'll be breathing a lot of this. And it has a bronchodilatory effect. It is a wide-spectrum antibiotic. Works on acne, it works on staph, works on Cryptococcus and Candida, and Campylobacter that causes a lot of epidemics of gastroenteritis. If we give it to mice, it increases their activity. It has a stimulatory effect. But what really is at the basis of this is an important attribute, that it's an acetylcholinesterase inhibitor. Acetylcholine, of course, is the neurotransmitter that is the memory molecule. And it is suppressed by THC. THC mean, where was I? That's the THC, short-term memory impairment, and that can be reversed. I don't have an excuse. That effect can be totally eliminated if there's pinene content of sufficient amount in the material. More recently, it's been shown that oxidative damage, like with hydrogen peroxide in the brain, can be, that this can be protective for astrocytes, the support cells for the neurons. Historically, pinene has been an antidote to THC overdosage. And then I love this. What do we do, particularly in the Northwest, if we need a break and need to clear our heads? Well, that calls for a walk in the woods. A walk in the woods here is going to expose you to pinene, and you're going to have a memory boost and probably a boost in alertness. And there's a term for this in Japan, which is forest bathing, or Shinrin Yoku. So I think what I'm going to prescribe for the crowd is a little Shinrin Yoku, if not today, then tomorrow. Now, much more prevalent, and the predominant terpenoid that's going to be found in cannabis of commerce is this, beta-myrcene. Now, it has some good attributes. It is anti-inflammatory via PGE2, but it's very sedating. And this is what I like to call the primary couch lock factor in cannabis. That's in combination with THC, where the person just gets immobilized. Some people like that. A lot of people do not. It's not necessarily good if you're a medical patient that needs to work or study. Interestingly, it is an analgesic, and the effect is blocked by naloxone, the same thing they give to people who overdose on heroin, mu agonoid antagonist. And so it has a very narcotic effect. When people feel that sedation, chances are it's due to this molecule. And then there's some other things. It seems to be good for chondrocytes, your cartilage cells, could be used in osteoarthritis. It would seem to treat ulcers, and it could prevent some injuries to the brain from lack of oxygen, like in strokes. So not all bad, but right now, too much of it in too many chemovars. Again, better and not always available is this one. This is D-limonene. I realize there's too much on this slide, but I left it that way on purpose to show you the versatility of this molecule. We'll just emphasize a few points. It's a very potent antidepressant and immune stimulator, and that's in humans. They did a study in Japan where this was wafted into the air on a ward with depressed patients who were able to cut their Hamilton depression scores in half, get off of conventional antidepressants, and improve their immune status. It has a bunch of other effects in reducing neuropathic pain. It may be effective in breast cancer. It's anti-inflammatory. And another one that's being actively researched is it's an agonist at the A2A adenosine receptor and likely will synergize with both THC and CBD, and that's an area of active investigation right now with my colleagues. At the bottom, too much detail, but we've got two kinds of fat, white fat and brown fat. Brown fat is what you use for energy. White fat is what many people have too much of. Limonene can actually turn white fat into brown fat and be helpful in treatment of obesity. So that's something that needs more work as well. Linalool is going to be familiar to you as the main component of lavender. And it's present to varying degrees in cannabis, sometimes predominant. Very powerful anti-anxiety agent, but with less sedation than, say, a benzodiazepine. Very powerful local anesthetic. You should probably, if you cook, keep a bottle of lavender essential oil in the fridge. And as soon as you get a burn, put it on neat, cover it with ice cube, and in 10 minutes the pain will be gone, and chances are you will not blister or have any other problems. It's really amazing. But this is also a very powerful anticonvulsant. Now, with the ability to analyze for terpenoids, my colleagues have reported that they're seeing much better results in seizure treatment in children where there is linalool in the material. Ocimene is also going to be familiar to you, at least to your nose, if you ever eat pesto in the summer. This is in basil. And it also can be a predominant terpenoid in cannabis. But we really don't know a lot about what it does on its own, particularly from a psychopharmacological standpoint. We know that it has some anticonvulsant and antifungal and anti-tumoral properties, and apparently it's important to honeybees and how they regulate their social lives, which are very complex. So that one needs work too. Another one that's quite common on West Coast cannabis is terpinolene, and if you want to know what that smells like, pick up a turnip, which makes a good salad, by the way. This reduces LDL oxidation, so it could be very important in helping reduce cardiovascular disease. Interestingly, although it's sedative in mice, the reports we get from people who use cannabis that has a high amount of terpinolene is a more stimulatory buzz. So there's obviously a difference between species here. And then it also has pain-killing properties. So this is quite common in some chemovars and deserves a closer look. A little less common is this one, delta-3 carine. This one is in wood. So if you have freshly cut wood, you're going to get carine as well as pinene. It's readily absorbed and metabolized. Interesting one with it is it seems to stimulate bone remineralization. It stimulates osteoblastic cells, so it could be used in osteoporosis. It's also interesting because in so-called sativas, it seems to be present. So it's a marker for that if you believe in these differences. Now we're going to turn to the 15 carbon molecules. These are the sesquiterpenoids. And in concentrates, or things that have been extracted, particularly with CO2, these often end up as being predominant. The most prominent is beta-caryophyllene, a fascinating molecule because it's not just a terpenoid, it's a cannabinoid as well. It was known for a long time that this is a very powerful anti-inflammatory. It's comparable to phenylbutazone, an anti-inflammatory that's used in horses but considered too toxic for people. But unlike it protects the stomach from ulcers. So about 10 years ago, my colleague Jörg Gerich figured out that this is a CB2 agonist, very specifically, a full agonist. And so a lot of its anti-inflammatory and analgesic effects are being mediated that way. It also decreased cocaine administration in animals, and it's good against some things like vectors of malaria. And it has a bunch of other effects that are in an excellent review of about 25 pages that came out last year, Sharma. Its close counterpart is caryophyllene oxide. I'll just mention this, the primary interest, it's in lemon balm. But two things about it. It seems to be the molecule in cannabis that leads dogs trained to find it, sniffer dogs. And then this one, you're spared from drug ads in Canada, but in the States, on TV, they have ads for this stuff for your nail fungus. To treat one nail for 6 months costs $3,000, and you've got a 50-50 chance that it's going to work. God forbid, you've got nail fungus times 10. But let's compare that to this. Caryophyllene oxide in a concentration of 8% knocked this out in 2 weeks. So which is better? But this is from a plant. It couldn't possibly work. Another common terpenoid in cannabis is this one, humulene. Let's be very familiar. The aroma, it's part of what's in hops. But despite being such a common molecule, almost nothing has been done on its individual pharmacology. It's important to fruit fly mating, which we won't go into. And then I found this one just last week. It prevents damage from hydrogen peroxide to rat astrocytes, reduces that 50%, and it concentrates within those cells 7-fold. So this is probably an important neuroprotective effect and could be anti-aging as well. Guaiol, I'm just going to mention briefly. This one is particular to Afghan chemovars of cannabis. It is a sesquiterpenoid alcohol. These molecules in general tend to be sedating, and it might be responsible for some of those effects. It also is a component of this that's used as a smudge or incense-like. So we've got a lot of these that end up in sort of the meditative context. And that may have, it might be the dreamier, more introspective types of cannabis that have these. And the same would apply to this, beta-elamine, no oxygen, but it's a component of myrrh, of frankincense and myrrh fame. I purposely left this slide way too busy because it may be interesting for you to know that this is an approved drug on its own in China. It's been used extensively to treat cancer because, like the cannabinoids, at high doses it kills cancer cells and it's not bad for normal cells. There was the study in 2013. They showed high reported efficacy in a number of types of cancer, including brain, leukemia, lung cancer, but not stomach, which is common in China. Probably because of too much carbon in the air, etc. It also reduced damage to blood vessels from hydrogen peroxide. There probably isn't a lot of this in most cannabis chemovars. However, because of the likelihood of synergy with the cannabinoids, it really should be something for which we should be selectively breeding, I believe. Now, we come to the cannabis odds and ends. I'm just going to take a quick time check. And you'll see why I call them this in a minute. We have, in the prairie provinces, hectare after hectare of hemp growing, for the fiber. And there's a lot of cannabis grown indoors as a drug, but there are a lot of roots, tons, metric tons of it being thrown away. Well, these have been used for thousands of years medicinally, going all the way back to Pliny and before. And there's some really interesting pharmacology there. No cannabinoids at all. But what you do have is triterpenoids, which are actually the most common phytochemicals in plants. And again, too much here, but you've got a lot of interesting effects here, a wide variety of anti-inflammatory effects and hepatoprotective effects, antimycobacterial activity for TB, also protective against ulcers. This stuff is available in these roots that are just being thrown away, and that situation needs to change. I know a few people, including in the audience, that are interested in investigating this. And a closely related one, epifridelenol. And again, this has been interesting, suggested as a dietary supplement or use in cosmetics. And there are some people that are making artisanal preparations that use the cannabis roots. For a century, people were looking for alkaloids in cannabis because they knew that they were in all these other plants like morphine, other opioids, etc., and they could never find them until they looked at the roots. And there is one unique to cannabis called cannabisativine, but its concentration is very little. On going to the source, I asked Dr. Mechoulam about it a couple of years back, and he said they are present in minuscule amounts and are presumably not relevant to any cannabis biological activity. Again, there's a graduate research project for you. On its closely related molecule, anhydrocannabisativine. Same thing, very low amount, but interesting for purposes of a presentation. One I suspect you haven't heard about is canoprine. This is an isoprenylated bibenzyl. It's present in various parts of the plant, particularly the leaves. It can be as much as 0.2%. It is also anti-inflammatory, inhibits 5-lipoxygenase and the COX-MPGS pathway. There's some closely related compounds that don't do that. What's funny about this is how much is there doesn't relate to how much of the cannabinoids there are. But if there's a lot of canoprine, there won't be much canflavin A and vice versa. There's a reciprocal relationship, and we'll talk about that one shortly here. I wanted to briefly touch on hemp seeds. These are grown in Canada and then subsequently imported into the US. We can't grow them legally. But again, on the desert island, if you had to choose one vegetable source food upon which to survive, this would be a good choice because it's very complete. It has all the essential amino acids. It's 35% protein in the digestible edestin, unlike the indigestible genistein of soy. And it has 35% of this oil rich in essential fatty acids, linoleic, linolenic, and gamma-linolenic acid, which you'll find in hemp seeds, in evening primrose, in borage seed, and in mother's milk, and about nowhere else in nature. and you can't make it on your own. And then there's canflavin A. I feel particularly attached to this because it was discovered by a friend of mine, Marilyn Barrett, about 30 years ago now. It was her doctoral thesis. So this was found in cannabis. It's a flavonoid, unique to cannabis, and it's 30 times stronger than aspirin as an anti-inflammatory. It's somewhere between aspirin and dexamethasone in that. So that was reported, and then nothing happened for decades. Subsequently, a couple of years ago, this was looked at, and they found out that you can make this in hemp sprouts at a stage where they're making canflavin A, and they're not making any cannabinoids. So that might be legal in all 50 states someday, not now. Anyway, this has a really interesting profile. It suppresses prostaglandin E2, the primary mediator of inflammation, and it also directly inhibits something called MPGS1. It doesn't affect COX-1 or COX-2. So this dual inhibition is felt to be an ideal profile for an anti-inflammatory, and it shouldn't have any side effects of note. And then, interestingly, again, something that's often thrown away, the hemp seed hulls. They have two compounds in them, cannabicin B, an antioxidant that also prevents LDL oxidation that's involved in atherosclerosis, and also seems to work on hepatic carcinoma. And then this other one that we don't know as much about, but has some similar effects. Now, if we're not too tight on time, I want to spend a couple of minutes showing some examples, and then we'll open it up to questions. We have the phytofacts here. I wanted to show the profile of some chemovars that are interesting. So this is a type 1 plant. It is predominant in THC, very high in pinene, very low in myrcene down at the bottom, so it will not be sedating and should not produce short-term memory impairment. So if someone needed THC for pain control or spasm control, they could take this and probably still work or study. This is a type 2 plant, Fruity Pop. I'm not fond of some of these names. Type 2 means there are roughly equal amounts of THC and CBD, and as you see from the colors on the bottom, it's very rich in several terpenoid components, so probably has very interesting effects. Another type 2, Sweet Surrender, roughly equal CBD and THC, high in caryophyllene, relatively high in limonene, again, general purpose with low sedation. Creamsicle skunk, type 2, this is said to be inspirational, and it's because it's got a lot of limonene and it shouldn't be too sleepy. So this will add to the presence, the uplifting effect that some people seek. Another type 2, Blue Dream, this is balanced with respect to THC and CBD. Again, very high in alpha-pinene, not too much of other things, good for work or study. This one's really interesting. More CBD than THC, high in limonene and beta-caryophyllene, moderate in myrcene, probably good. It's very high in linalool, I should say. This is likely going to be very helpful for burns and for epilepsy treatment. Two very different things, but the profile would suggest that. Let's see. This is called lemon CBD because of the high limonene, and it's a type 3, almost all CBD, very little THC titer. On type 3, this one is high in CBDV, cannabidivarin, as well as CBD. So it should be very good at treating seizures, also because it's high in linalool and has some anti-inflammatory effect from caryophyllene. And I don't know that anyone, this is very new, I don't know that anyone's used this for epilepsy treatment, but it would be a great candidate. And I'm going to stop there and see if we have time for questions.
Thank you so much. So we are going to open it up for questions from the audience. There is a microphone.
So I was just curious about all the anti-inflammatory molecules in cannabis. Is there any purpose for the plant, for that to benefit the plant, or is it just a metabolite like a lot of other compounds?
Yeah. If you're really interested in this topic, I'd suggest a book by Kennedy, Plants in the Brain, that goes into a lot of the ecological reasons that plants make these compounds. Often they're protective for the plant in some fashion as antioxidants, so if it gets wounded by insect predation, it can heal. And, we're real different than plants, but not so different that some of these effects can't translate over to being helpful to humans. And then you get discrepancies, things that are toxic to the plant may be salutary for us. But generally, I really appreciate the question because there are ecological reasons. A lot of these things are metabolically very difficult and expensive for the plant to make. Nature doesn't waste energy, and if they're there, they do have a purpose. It's just that it's only recently that there have been scientists interested in the why of these and the how of how they're produced. And then the next step, which is how do we leverage this into therapeutics for humans. But there's always a reason. Oh, now don't be shy. Up here.
Dr. Russo, thank you very much. How far are we away from being able to test somebody's endocannabinoid tone so we know what they're short of and what they maybe got an issue with?
Yeah, thanks for that question, Hutch. It's clear that there are a number of diseases, particularly difficult to treat pain conditions that seem to be associated with what I call a clinical endocannabinoid deficiency. Premier among these would be fibromyalgia, irritable bowel syndrome, and migraine. And with migraine, it's been shown with cerebrospinal fluid, spinal taps, that during a migraine attack, that the levels of anandamide go way down. Doing a lumbar puncture is invasive. The reason I didn't do that study in the States was I couldn't get it by an ethics committee, I'm sure, but they did do it in Italy. We don't have the ability right now to do scans in live patients to show what the density of the CB1 receptor is, what the anandamide or 2-arachidonyl glycerol levels are in the brain. But again, if we had that, we could really have a better idea of what ails people and what kind of cannabis-based medicine they might need to treat them. So hopefully that's in the future. We're not there yet, however. This lady up here.
You mentioned something about how it reduces cocaine or something, the caryophyllene?
Caryophyllene,
Caryophyllene.
Right. Sure. Okay, so that's mediated through the CB2 receptor. Most of the work has been on other molecules. Now, this always happens, and it amuses me because of my iconoclastic nature. But there are a bunch of people researching synthetic CB2 agonists to treat addiction. Now, what's happened so far is they haven't gotten anywhere because, again, the idea is let's design a molecule that has the highest affinity and potency at the receptor, and then later you worry about whether it's toxic. And almost invariably it is. Instead, we've got this natural molecule. It's in the peppercorns on your kitchen table. And it does exactly the same thing, and we know it doesn't have any serious side effects. But unfortunately, profits would dictate that you make your own molecule and have intellectual property rights to it. And God forbid you should use a natural molecule that somebody else can also use and you won't make any money on it. Terrible thing. Sir.
A recent question. Since January the 20th, there's been a little bit of upheaval out in your country. And yet on the one side of it, you've got the legalization in a number of states relevant to recreational use and the rest of it. And yet, just 5 minutes ago, you mentioned about how you could not take and do certain research in the United States, and so it was done in Italy. So do I sort of interpret that there's a divergence of paths here where recreational, and Canada, of course, is going down its separate path? Whereabouts is the research going to take and be done that is from a medical perspective that you could not take and do last year in the United States? And you definitely will be, I assume, much more restrictive next year in the United States to do anything along those lines. So whereabouts is this, and then plus you got all the big European drug companies coming down on like we don't want none of this stuff, okay?
Right. Well, I don't know, short of having a crystal ball, and anybody who claims to know what's going to happen is probably delusional. No, it's a serious problem. I worked for a foreign company for 11 years just because of this problem, and it may be that I have to again. So I can only laugh before I cry. I don't know. I think the signs are ominous for biomedical research in general and in this area in particular, but we need to see. I don't know what's going to happen.
Thank you.
Sure. Up here, sir.
Sorry. From a pharmaceutical perspective, the FDA and most drug authorities worldwide are more puristic. They like one thing in their medicine compared to multiple of things, making it difficult to get approvals. Is that something you feel is a big hindrance to marijuana or cannabis becoming mainstream use in medicine?
Right. It's not a total blockage. Keep in mind that Health Canada approved Sativex, a whole cannabis extract pharmaceutical, in 2005, okay? And I expect Epidiolex, which is again a cannabis-based medicine, to even be approved in the United States later this year. There is a blueprint for producing a pharmaceutical from a botanical, plant-based medicine. It requires the same standards of safety, efficacy, and consistency that a new chemical entity, a synthetic molecule, will require. The difference is in demonstrating what all the components are, the hundreds of peaks that you'll have chemically in a botanical, and that they're consistent over time, and that's the challenge. However, it has been done, it can be done, there is a blueprint for it, and Health Canada will approve other botanical medicines if someone has the time, energy, and capital to research it. Please.
Thank you so much. Can you say a little bit more about the possibility of CBD converting to THC and just the discrepancy that people...
Sure. Yeah, I'll repeat the question. It was again on the bioconversion of CBD to THC. I should mention that if someone tries to decarboxylate their cannabis at too high a temperature, they will convert some of the CBD to THC. What I'm saying is your body cannot do that, okay? But I know of so many instances of people that buy something that's supposed to be 1 THC to 30 CBD, and they extract it and have a terrible experience. Either they're have orthostatic hypotension and pass out, can't get up for 4 hours, or freak out because of THC content. So we don't know what the optimal approaches are. It is possible to decarboxylate at a slightly lower temperature, for example, 90 degrees Celsius, but it takes several hours for that. And in the meantime, you are losing the volatile terpenoids as well. So anybody that thinks there aren't sufficient research projects in growth and preparation of this plant, oh boy, there's tons of stuff yet to do.
Just to follow up around that, because there's a lot of anecdotal experiences. I've also had this experience where I've used CBD and had a coffee and then definitely feel a high similar to THC.
Right.
Just wondering what your thoughts are on that and whether there should be disclaimers around, especially for the elderly.
Well, in fact, elderly people tolerate cannabinoids very well. Yeah, I mean, it's always wise to be cautious, but for example, in the Sativex development program, there was a 98-year-old guy who was on massive doses and he had no side effects. So yeah, it can be done, particularly with the slow titration. The psychoactive effects of THC are what are responsible for the situation you describe. And my explanation is that somehow or other there was too much THC. But starting extremely slowly and titrating upwards as needed just to control symptoms, not to the point of psychoactivity, that's the proper approach to cannabis therapeutics. And again, there is this difference. Most symptoms that cannabis treat do not require attendant psychoactivity. Now, I'm not going to tell you that euphoria is a bad thing, although the FDA considers it a side effect. But it absolutely is the case that most patients can attain pain control, spasm control, control of bladder function at a dose that isn't interpreted as intoxicating. And for most people, that's what they want. They want to live a normal life with control of symptoms. If they want to get high, there are plenty of ways to do it, but it's not a necessity. Sir.
Given that certain pharmacological agents change the remodeling of proteins after long-term usage, do you know if there's been any work on whether or not any of these compounds change the expression patterns and levels of various proteins involved?
Yeah. Yeah, it's a big area. There are two things I'd mention. I have heard it reported that CBD produces methylation of DNA, but what the practical implications of that are, I cannot tell you. Then, a lot of these molecules will affect expression of proteins, particularly in tumors, in cancer tumors. But in terms of being mutagenic, changing DNA structure that produces mutations, they're all extremely safe, everything I've presented to my knowledge. But yeah, absolutely, there are differences because of these molecules in the protein expressions, absolutely. What that means, you'd have to look at each individual instance. But there's plenty of literature that includes that. And it's at this point I remind people I'm just a neurologist. I'm not a pharmacologist or molecular biologist, but there are people that are, and they have reported these changes.
Okay, we've got lots of questions. I think we'll take two more. And then Ethan's going to be around for the reception. I would encourage you to stay for a few minutes because there's a lot of food that needs to be consumed outside. So let's take two questions. I know there's one there and there's one. Yeah, go ahead.
Yes, I'm going to ask you to look into your crystal ball. There's a lot of excitement right now, and there's a lot of exciting research on cannabis because it's been identified for all these medical uses. in Canada, we're moving towards recreational use. In your opinion, do you think going recreational is going to encourage more research or discourage the research? Because now people can self-medicate and choose and decide what works for them.
Right. Well, I hope it's the former rather than the latter. However, there's some cautionary tales. We have legalization in Washington State, and from my standpoint, it's been a disaster for patients because of the lack of availability of previously available chemovars, for one, and an inadequate quality control. And I'm going to point the finger at the people that were told differently and didn't do enough. I wrote a paper this year that's published. We looked at 26 legal cannabis products bought in Seattle and sent to a state lab, and looked at pesticide exposure, and there was gross contamination in 86%. And it had been recommended by the consulting group that they mandate testing for pesticides, and it was not done. No matter how good things are, they can always be better. So I do hope that there'll be more research, but I don't know where it's going to come from. It won't be from the National Institutes of Health in the States. They're going to be gutted. I'm hoping that Health Canada will kick in and the private industry will pick up some of the slack. There are a lot of people interested in this, and if we get some entrepreneurs and people that might have made money in other fields that are interested, I mean, look at what the Gates Foundation, they're not seemingly interested in this topic, but they've been great for other biomedical research. So hopefully there'll be enough interest that will lead to funding. That's our only hope.
We'll take one question in the back there. Yeah.
You mentioned there was stimulation with serotonin and anti-inflammatory effects with some of the molecules. I was just wondering if there was any connection that was solid enough for common problems like rheumatoid arthritis, AD or ADHD.
Yeah, that's an odd one. In attention deficit hyperactivity disorder, there are a lot of paradoxical effects of drugs. So stimulatory drugs seem to calm activity down. Drugs that are sedating for other people may lead to increase in activity. When I was in practice, I had a number of adolescents and adults who independently, without knowing anything, discovered that cannabis, which would have been predominantly THC, helped with their ADHD a lot. There have been 2 studies in animals using animal models of ADHD showing benefit of THC. And then certainly other components, the terpenoids, could be helpful. But I don't see anybody rushing to do a clinical trial there. Obviously, for a common disorder of extreme importance from an economic and public health standpoint, it deserves proper attention, but it's going to be a very hard thing to study. The idea of giving cannabis to kids for any reason is anathema to a lot of people. I would submit to you that there's no drug that's been developed in adults that hasn't found its way into pediatrics. I'm a pediatric subspecialist. Cannabis was used extensively in kids for a century between 1840 and 1940 when things went south. But they didn't seem to have any worries about it, and the species hasn't changed so much since then. Much as we'd like to think that we're so much more evolved, it isn't true.
Okay, so we'll stop here, and as I said, there are things happening outside. If you could just hang in there for a few more seconds, Ethan, I'd like to present you with this gift from the committee.